Hello, and welcome to the single molecule localization microscopy image analysis workshop as part of the I2K Image to Knowledge conference in 2022. My name is Thomas Bengal from the University of Minnesota, and this workshop is co-organized by Sean Colley and Christian Frank. After a quick overview of the idea behind this workshop, we'll dive into presentations. We have 3 presentations each, 10 minutes, which will be part frontal presentations and part demonstrations of the associated software. So a single molecule localization microscopy enables us studying biological structures beyond the diffraction limit. It has the unique ability of capturing spatio-temporal coordinates for individual molecules. But beyond imaging, it is increasingly used as a quantitative approach for molecular clustering, tracking, and stoichiometry. Analytical tools are therefore essential components at all stages of a single molecule localization microscopy experiment, from building the multidimensional pool of data points to the analysis of that pool to generate high-resolution data. So we saw this as an opportunity for a focus issue on the topic. And in particular, this is a research topic in Frontiers in Bioinformatics, Computational BioImaging section, which was recently finished after about a year and a half of work. We have 12 articles as part of the research topic, so been very lucky in terms of participation. 2 of them are technology and code. We've got 5 original research articles, 3 methods, 1 review, and 1 opinion article. The research topic has had a good success, about 17,000 views in total. 13,000 of them were inside each article. And we had about 1,594 article downloads for a total of 2,000 views of the topic itself. So without further ado, let's dive into the presentations.
Hello, my colleague Angel and I are presenting the software behind our paper, Efficient Cross-Correlation Filtering of 1- and 2-color single molecule microscopy data. In this work, we will discuss the importance of co-localization in microscopy, how single molecule localization in microscopy can help aid in studying co-localization and protein clustering. We'll then discuss the existing techniques used to study co-localization using single molecule microscopy datasets, specifically pair correlation and cross correlation, and we'll discuss the limitations of those existing techniques. And then my colleague Angel will present our method and a code walkthrough so that you all will know how to use the code. Protein interactions and protein organelle interactions are a hallmark of cellular signaling. Studying proteins that are close together, each other, or proteins that are close together organelles can help decipher how those proteins help impact cellular functions. And by quantifying the degree of clustering of a protein in the cell, or how quantifying the degree of co-localization between 2 protein populations can gain insight into various cellular functions, especially if you start changing cell states. Single molecule localization microscopy technique is a relatively recent technique that is able to take the measured intensity profiles, fit that to a model function to find the center of that protein to a very high precision. And typically, people use this by getting single molecules across a very long period of time, then reconstruct a cellular structure at a very high resolution. But one of the unique things about this technique is that you can get the actual coordinate positions of each single molecule inside a structure, which makes it very easy to quantify the degree of co-localization or clustering. A technique that's typically used to quantify co-localization and clustering in single molecule microscopy datasets is the pair correlation and cross correlation functions. These functions take the pairwise distances between a single color dataset. So they look at all the molecules and calculate all the distance pairs between those molecules, or in cross correlation, calculate all the molecules between two different datasets, and then use that to then calculate a cross correlation curve that's able to quantify the degree of clustering at specific distances. And we can use this cross correlation curve to then find a distance where we say that molecules are co-localized and not co-localized, and then we can use that to separate co-localized populations and non-co-localized populations, or separate populations. But one of the challenges is that calculating this pairwise distances across the entire field of view of mammalian cells is very memory intensive. The goals of our approach are, one, to be able to calculate this distance-dependent correlation functions across the entire field of view of a camera with very limited memory cost. And then we want to use this to identify and separate clustered and co-localized molecules from separated and non-co-localized molecules. Now my colleague Angel will discuss the details of our method.
Our approach is to use a range search instead of an explicit distance matrix to decrease the computational complexity of storing the distances. So we only consider co-localization up to some distance away. We store the distances in a k-dimensional tree, and then we construct the cross correlation out to some distance. Then based on the cross correlation, we determine some threshold distance to then apply to splitting the molecule list. That is, splitting the molecule list into co-localized and non-co-localized. By doing this, we can then perform other secondary or post-analyses like clustering, counting, other pair correlation, linear distribution analyses, structural analyses, and diffusion analysis. Here we show the results on simulated data, which is provided also along with the code. So we have all localizations combined here, which consists of ground truth with noise added. This noise consists of single localizations as well as these large clusters that don't co-localize with anything in the ground truth. And then applying the method, we can recapitulate the isolated clusters, which corresponds to the ground truth with some additional error, as well as the other localizations, which consist of mostly the noise and the large clusters. Here we show the radial distribution function, which is used to determine the threshold distance for the clustering that we performed before the co-localization. We also show the cross-correlation function that we use to compute the distance for determining non-co-localized and co-localized. Here we show the distributions of molecules per cluster, as well as the radii of the clusters. And we show in green circles the ground truth, and in red X's the recovered molecules. And we show that we can recapitulate the original ground truth to high accuracy and exclude the noise. Using a range search to correlate the molecules increases using a range search to correlate the molecules uses an order of magnitude less memory than the explicit distance matrix without incurring a significant overhead in the elapsed time. The code published in this paper is also available on GitHub at this link. You can just click the green code button to download the code along with the simulated data. Now I'll show an example of running the code in MATLAB. So first we set up a sort of preamble where we load our data and we format it for using it in the code. The point of entry in the code are these two functions, CC graphic pipeline and CC separation pipeline. Graphic pipeline computes the pair correlation and cross correlation and gives some reasonable distances for using as cutoffs in CC separation pipeline, which then returns a separated molecule list based on the supplied parameters. So we'll run the preamble. Next, we'll run the graphic pipeline. And this generates 3 plots. The pair correlation for the rate of distribution function for population 1, and it shows the 90% cutoff of the fall from the peak. Then it shows rate of distribution 2 with the 90% cutoff from the peak. And then it shows the cross correlation with the 50% cut distance and the 90% distance from the peak. The pair correlation cutoff distance can be used for pre-clustering, which we perform in this case. And the cross correlation cutoffs can be used for the molecule separation. So in the next step, we supply these parameters to the CC separation pipeline, and then we run the code. And in this case, we write the files to this directory here with the results. So we have, this is all of the data that's in the figure of the paper. And then here is the separated data, the excluded localizations and the included localizations. This can then be compared to the ground truth to determine how well the ground truth is recapitulated by the analysis. To conclude, we present the cross-correlation method that can be used for molecule filtering across large fields of view with the added benefit of not being memory intensive. We also, in the paper, compare the performance to other colocalization methods. And here's the DOI for the paper if you want to look at it. And then the code is also available in the GitHub link supplied.
Hello, and welcome to this workshop about our recent publication, Raw Data to Results, a hands-on introduction and overview of computational analysis for single molecule localization microscopy. As an introduction, I would like to give a very brief overview on single molecule localization microscopy and how it surpasses the diffraction barrier. Now, from a sample point of view, what you require for SMLM is reversible fluorophores. So fluorophores that can basically be turned on and off roughly at will. And there are more or less 2 options in there. First of all, fluorescent proteins, such as this photoactivatable GFP, where there is the chromophore inside the barrel structure is being formed by being photoactivatable, which means that by inducing this photoactivatable change, you can induce how many of your fluorophores are turned on or off. Similarly, you can do this with organic fluorophores. For example, this the cyanides, I think here is size 7 at the bottom, where the addition of this sulfur group makes or breaks the conjugated system and thus induces fluorescence. Now, with this blinking fluorophores, you basically can do subdiffraction limited localization of individual molecules only because you are 100% sure that these are indeed individual molecules. So the data analysis, the first step is always that you have a whole movie of individual emitters. You localize them with subdiffraction limited precision up to 20-30 nanometer precision. And with that, you can in theory do 2 main routes, either super resolution imaging or single particle tracking. And in super resolution imaging, you basically have an unknown structure before you, this ring in this case, where you have a lot of blinking emitters, and by imaging and localizing them one by one, you can resolve the original structure. Alternatively, in single particle tracking, you can follow individual molecules through space and time and look at how big they jump, and from there do quantitative analysis. Now, the problem is really that if you have a movie like this, And especially for experimentalists, if you have a movie like this, you want to go into the data analysis and to really dive deep into the data analysis. But then to really know what you need to do there is very overwhelming, because you can do imaging, right? And you need specific algorithms and specific computational processes for that. You can do single particle tracking, where you actually need a slightly different toolbox of sets, even though the beginning might be the same, but you run into different caveats. And you can do all kind of interesting mathematical quantitative analysis, such as here, I determine localization precision. But overall, if you're not used to do this kind of data analysis, for example, if you're new to a lab or if you're new to this technique, this is just very much a big black box. And our manuscript was a way to circumvent this black box. So what we created is a sort of easy to understand and adaptable code to have the full procedure for SMLM, from raw video data all the way to full data analysis and interpretation. Now, we focused on an understandability of the code rather than having the best speed or the best quality. And therefore, we provided well-documented code. We have pseudocode for every module that we provide. I will go over an example in a bit. And everywhere where needed, there is example data provided, so you can just run the code and see what it does and see how it works. Alternatively, we discuss the best software packages that you could use in your day-to-day analysis, which do the same, roughly the same protocols and the same algorithms that we provide in our manuscript. Finally, all of this code is available in multiple formats. Most importantly, it's available online, freely accessible on Google Collaboratory, which means that this is a Python written code that you can easily copy and tinker with in the browser itself. So you don't need to install any code bases, any software, anything. You just can run it directly from the browser. Now, we split our whole code base in so-called modules. So we have 11 modules here, and we split those modules again in 3 different steps. So the first one is a pre-processing and localization, where we go from raw movie data to a list of localizations. Then there's a step of post-processing options, where we can go from original localization data to corrected localization data, where, for example, you have localization merging or drift correction. And finally, there's the data interpretation step, where you go from this localized data to actually good interpretable results. You can think about image generation, single particle tracking, or localization precision. Now, as an example, I can go over one of the modules. So if you follow our manuscript, you can come to this module's overview page on Google Colab. And here it's repeated again, all modules, and here is a link to every module. Now, to take an example for module 2, the localization of single molecules, I have it opened here. What you will be greeted with is a page like this. And this is a Google Colab page where there's a mixture of images, texts, and code that you can directly run into the browser. So here we start with a simple explanatory image of what we're going to do. Here in this cell, we're going to download some required data for this module. And then we can follow step by step the whole procedure to do full localization from raw data to localized points. Then the first step we have is a sort of identification of regions of interest with a so-called difference of Gaussian filtering. And as you see here, you really, the code here is really written line by line, well documented. And if you want to run it, we can press this play button here. And you see that it runs this cell and it shows this output. Now, maybe we want to change something here. Maybe we don't want to look at frame 30, but we want to look at frame 35. And if we do that, we see it's directly adaptable and we look at a different frame now. Good, then the next step would be to actually find a region of interest where the emitters are residing. And this is done with a difference of Gaussian filters. So here we apply 2 different Gaussian filters and we show the image of actually the difference of the 2 Gaussian figures, right? And here we get this image. Then the next step here is to quantify the region of interest, finding the maxima. And to do that, we can simply find sort of the twice the standard deviation and look at where local peaks are found there with this script. And with that, we can simply show here with the PLT scatter command every single emitter that we find with a small circle. Then we can isolate these individual regions of interest to simply cut out a small 7 by 7, I believe, picture around it, and there we can see the individual emitters. Finally, we can do subpixel localization, so we get our localized point where our emitter is with subpixel position, and we do this with a phaser fitting manuscript because it's fast. And then you see that now for every of these images of these point spread functions, we found the exact center with the exact coordinates. Then we do a small intensity routine based on photometry intensity. So we can order them from low to high intensity. Finally, we declare this all as functions. It's simply the same code as above, but now declaring as functions, also to explain how functions are written, what their syntax is, etc. Which means that in the end, we can, of course, do the complete localization procedure, where we now have simply the function that we created on our single frame, which means that we can get all our XY intensity information out from the single frame. And of course, we can easily multiplex that to the whole data. Now, this is roughly what you would expect from a single module. In addition to this Google Colab, where you can play around with, tinker with, look at the code, and just run it and understand it. There's also in the supplementary info, there is a pseudocode corresponding to every module, where, for example, this is a supplementary pseudocode corresponding to the module that we just had, where it's in very easy layman's terms explained exactly what we're going to do without going over any details, right? We're just going to say, okay, we're going to calculate the difference of Gaussian frame to enhance the features, rather than really talk about how we're going to do it. Finally, if we go back to the modules overview, you can simply follow every of these modules. There's also a supplementary code snippets module, and that's just a very small overview of different quick snippets that might be interesting to look at. For example, filtering localizations, changing your ROI, or changing localization units and things like that. Now, if we, finally, what I want to tell is that all this code is also available in MATLAB format in our supplementary data. And finally, I'd like to thank some people. First of all, the co-authors, Ulrike and Bartus, and 2 testers that we had, David and Laura. And of course, I'd like to test everyone from our AG Endesfelder group who are now at the University of Bonn. And thanks a lot for your attention.
Welcome to my presentation about valid p-values for a single molecule microscopy. So first, what is single molecule microscopy? If we have some proteins in the cell, then we can label them with fluorophores and observe them with fluorescence microscopy. So the proteins are typically quite small, so in the image they actually should be visible as a small point only. However, we have diffraction, and thus we have a point split function with a width of around 250 nanometers. However, if we only have one fluorophore, then we can determine the center of the point split function, and for this we have quite small localization errors. So one application of single molecule microscopy would be single particle trajectories, where we can either track the position of the molecule or some other feature like the brightness. One other application would be single molecule localization microscopy, where we have a very small structure, and in a diffraction limited image, all the point functions would overlap and we would only see one blurry spot. However, we can separate the fluorescence of different fluorophores in time and then determine all the positions and combine them to the final localization map. So now about p-values. P-values are very important as they are the basis for significance testing. And for a significance test, you need a certain test statistics, and under the null hypothesis, this test statistics will show a certain distribution with likely ones and rather unlikely ones. So if you record an experiment, then you will observe a certain value for this, and the p-value is just given by the integral under the curve here. And importantly, if the p-value is valid, then it should show a uniform distribution under the null hypothesis. However, two common pitfalls are multiple testing and correlated data, where the p-value distribution will look more like this, with a peak at small p-values, leading to wrong and irreproducible results. So now we'll come to the first application, which is single particle trajectories, where you have a certain track. And here, for example, we recorded the thread efficiency. Just to shortly explain that, there you have two fluorophores, a donor and acceptor, and the brightness ratio between the two tells us something about the distance between them. So a colleague of mine wrote a very nice software package for a simulation of such trajectory, which you can find under this link here. However, within those trajectories, we have inherent correlation of the data. So you can correlate a trajectory and other ones for a certain group A and another group B. And then you can ask yourself the question whether there is a significant difference between the two groups. You can answer this with a permutation test, where you take all the data and split them up randomly into new groups A and B. And then you record or you calculate a certain test statistics. So here we just took the difference between the means. And in the end, you can compare the test statistics as zero you obtained for your recorded data to all of those from the permutations. And based on this, you can calculate a p-value. However, if you do it like that, due to correlation in the data, you will get a very pronounced peak at small p-values. So what can we do to avoid that? What we propose is a block permutation test, which keeps together the data which belong to the same trajectory. And if you do it like that, then you will obtain a valid p-value. So for this, we also provide some software. So you can go here to this link and to the subfolder permutation tests here. And then you will find a function permutation tests. You can just put in your data for trajectories A and group B, then select the number of permutations and whether you want to have the block approach or the standard one. And you can also set your level of significance. And in the end, you will get your p-value. And now I will come to nanoclustering in SMLM, where we have the problem that a localization map usually doesn't look like this here, but more like that, where you have multiple detections of the same molecule leading to overcounting and hence localization clusters. And you can also see that in cells, where here you have the diffraction limited and the localization map. And here you can see these little localization clusters, which actually you cannot see in the diffraction limited image. So the question is, are these localization clusters real molecular nanoclusters or not? So here you have the same problem again. You have the molecule positions which are randomly distributed and clustered. However, if you have a look at the localization maps, it will look more like this, where you cannot see a clear difference between the two. So what we came up is an approach termed two-cluster, where we take two-color SMLM localization maps, and then it looks like this. And here you can actually see a clear difference between the two, because here all the localizations come from the same molecule, and hence they are one color only. Whereas here within a cluster, you have multiple molecules with different colors, and here you have both the colors present in one cluster. And to assess this, we took the cross nearest neighbor distance, which is rather large in a random case and quite small in the cluster case. And then we calculated the cumulative distribution function of those distances. However, if you have only one function, you cannot tell much yet. So what we needed is some controls, and we generated them via toroidal shifts, where you take your recorded data and then shift one color channel. And by this, you split up the correlation between the two and get a random localization map in the end. So with this, you have your controls, and you can do this with different shift vectors, and in the end, get all these gray curves here. And for the random case, your original data, the green line, lies well within all the gray curves, whereas here you see a clear bias towards shorter distances. And based on these curves, you can also determine a p-value in the end. So to easily apply this method, we wrote an image shape plugin, which you can find under this link here. And here you can find a plugin, also a detailed manual, and here you can find a short quick guide how to install the plugin. If you have downloaded and installed the two-cluster package, then you can open Feature or Image Tray, go to Plugins, two-cluster, and run the analysis. And then this window here will open, where you can select the folder where your data files are stored. So here you can select the localization data for color channel 1 and color channel 2. And then here you can set some parameters if you want to. However, it is recommended to just stay with the default parameters. And then you can start the analysis, and you will be taken through all your data files. And for each one, you can set the region of interest that you want to analyze. And then you can go to the next one. And if you're done with all the data files, you can click Confirm, and you will be taken to the results window, where you see the parameters that you set, and you see the histogram of all the p-values that were obtained. And here you see the results for each individual file set with the localization data and the cumulative distribution functions of the nearest neighbor distances. And here with the arrows, you can browse through the file set. And if you're done, then you can just save the results to folder on your computer, and in the end, close the window. And moreover, we have a MATLAB version of the code, which you can find here and use if you want to, for example, adapt it. And now I will shortly come to the problem of multiple testing. So if you have multiple cells and you calculate a p-value for each, then it's quite likely that one of them will show a small p-value. You can also see it here, where we have random molecules and clustered ones. And also here, we have one small p-value. So in order to avoid this issue, we can calculate one combined p-value for all the experiments with this formula here. We just set a threshold p0. Then you take the number of overall experiments and the number of experiments with a p-value below the threshold. And then you can just apply this formula and you get your overall p-value. We also wrote a software for this. You can find it here under the subfolder multiple p-values. And this is essentially just this function where you put in all the p-values that you obtained and the threshold, and you will obtain the overall p-value. And with this, I'm at the end, and I want to thank all the people of the biophysics group and also the collaboration partners. And here you find an overview of all the publications that I mentioned, and also all the software. And if you have any questions regarding the software, please do not hesitate to contact me.
I'd like to thank all of the presenters for their presentations. And thank you for watching the video. And looking forward to seeing you all Q&A session together.